all the others are true, then IRE is a stem loop structure in a transcript, right? That IRP binding to a 3' IRE will increase protein expression, and that's advantageous because it increases the amount of transferrin receptor. So if we just recall the slide, in the absence of iron, this will be sensed by the IRP, which will have that disrupted iron-sulfur cluster, allowing it to bind to the IRE, which can be the stem loop either at the 5' or the 3' end of the transcript. If it's on the 5', this will block translation, so you can just imagine a ribosome would have a hard time getting through this IRE. So this would decrease levels of things like ferritin, ferroportin, or ALA synthase. And this makes a lot of sense because under low conditions of iron, you're not going to be wanting to export iron with ferroportin, you wouldn't want to store it in ferritin, and you wouldn't want to start synthesizing heme if you don't have enough iron. Under conditions of low, we can bind the 3' IRE, and this will increase levels of proteins like the transferrin receptor, which would be advantageous because we can take up more iron from the plasma, right? So increase the intracellular amounts. So 3' IRE, it stabilizes and increases protein. 5' IRE, it blocks translation, decreases protein. Kind of contradictory. Okay. Easier one. Vitamin B12 or cobalamin absorption occurs in which area of the GI tract? The stomach, the duodenum, the jejunum, the ileum, or the large intestine?
You cleaved it or something.
So this is correct. It's absorbed in the ileum. So if we remember, for vitamin B12, it traffics all the way down to the ileum for absorption. In contrast to a lot of our other macromolecular nutrients, which are going to be digested and absorbed in the duodenum, all the way down to the distal portion of the small intestine. And so another question on B12. So the absorption of cobalamin or vitamin B12 involves A, the reduction by duodenal cytochrome B, DCYP prior to transport, cleavage of polyglutamate moieties prior to transport, association with intrinsic factor for recognition by cubilin receptor, transcellular diffusion, or uptake via the reduced folate carrier. There's some long answers, so I'll give you a minute to think about this.
Because you associate with intrinsic factor cubilin. Vitamin B12 and intrinsic factor.
See?
See? It should be seen.
Yes, this is correct. So B12 requires association with intrinsic factor for recognition by the intrinsic factor receptor, also known as cubilin. So these other answers, reduction by DCYP or duodenal cytochrome B, that's iron, right? So ferric iron requires DCYP for reduction. Cleavage of polyglutamate moieties, this refers to folate, which can have the polyglutamate moieties attached to the end, requires removal of those glutamate residues. No transcellular diffusion, and then reduced folate carrier also has to do with folate, but it's primarily taken up by the proton-coupled folate transporter in the enterocyte. So for vitamin B12, we just remember that it's released from food in the stomach by proteases as well as the acidic environment of the stomach. Parietal cells are playing an important role by releasing intrinsic factor and acidifying the stomach. B12 associates with the R protein. The R protein is degraded in the duodenum, allowing B12 to associate with intrinsic factor. This goes all the way down to the ileum, where it's taken up and associates with the intrinsic factor receptor cubilin. It's taken up. The intrinsic factor is degraded in the lysosome, releasing B12. Right? Okay, so two more questions, I think. So tetrahydrofolate and cobalamin. This was the last lecture. They play important roles facilitating the conversion of homocysteine to methionine by transferring which
A methyl
chemical
Methyl
group?
group.
An ethyl group, a
One
methyl
carbon.
group, an acyl chain, a phosphate, or an acetyl group. Correct. It's a methyl group. So anytime you hear tetrahydrofolate or cobalamin, you usually think of methylation reactions. So we think of this cycle where methionine is converted into homocysteine and allowing this molecule SAM to play a role as a donor to methylate an acceptor, and that can be DNA for epigenetics, RNA, or proteins, right? And vitamin B12 is playing an important role because it can methylate homocysteine together with this enzyme to reproduce the methionine, so you can continue to have successful methylation. And then cobalamin or vitamin B12 can regenerate methylcobalamin with 5-methyl tetrahydrofolate. You get transfer of that methyl group. So one more. So B12 deficiency causes megaloblastic anemia because B12 is directly required during thymidylate synthesis, so during DNA synthesis, because B12 is necessary for the insertion of iron during heme synthesis, because tetrahydrofolate becomes trapped as 5-methyl tetrahydrofolate and cannot participate in thymidylate synthesis, or because B12 is required for erythropoietin secretion from the kidney. Okay. Okay, so the majority has it. This is correct. So tetrahydrofolate becomes trapped. So just let me remind you that in this cycle, we have 5-methyl tetrahydrofolate contributing to the formation of cobalamin, methylcobalamin. If we lack vitamin B12, so if we take this step out, then 5-methyl tetrahydrofolate becomes stuck. So we can no longer regenerate tetrahydrofolate from 5-methyl tetrahydrofolate because it requires this cobalamin for this step. So without it, we have this trap, and we refer to this as a folate trap, because even though we have enough folate present in the cell, it all becomes trapped as 5-methyl tetrahydrofolate, and we don't have enough THF participate in other important functions of THF. And one of those functions is in thymidylate synthesis, which is required for DNA. So this 5,10-methylene tetrahydrofolate is necessary for this step. But we no longer have enough 5,10-methylene tetrahydrofolate because it's all stuck as that 5-methyl tetrahydrofolate. Okay, so we call that a trap. And so it sequesters tetrahydrofolate and can't participate in this step of DNA synthesis. And so we get that megaloblastic anemia, which refers to large macrocytic anemia, because these cells have difficulties with DNA synthesis and they no longer divide. So they get these big cells. So you'll get this both with depletion of folate. So if you have folate deficiency or a deficiency in B12, you'll get megaloblastic anemias. And that's particularly due to this loss of THF that participates in DNA synthesis. Okay, so let's move on to today's lecture on vitamin K metabolism. We'll take some time to first discuss kind of the basics of vitamin K chemistry and diversity and its sources. We'll discuss the underlying biochemical function of vitamin K and then move on to its importance in physiology, particularly in blood coagulation, before discussing assessments and consequences of deficiency. So I'm often really interested in kind of the history behind these vitamins and the history behind biology, because I find it really interesting because there are people behind all of these different discoveries, and it's often informative to see how people stumble upon these discoveries, right, that we see in our textbooks. And so vitamin K was discovered by this Dutch scientist named Heinrich Dam in the 1920s, and he was awarded the Nobel Prize for this discovery in 1946. And it's interesting because, as with many of the breakthrough discoveries in science, Heinrich didn't set out to discover a new vitamin. He was studying cholesterol metabolism using chickens as a model system, and he sort of stumbled into this area. And so what Heinrich was doing was he was feeding chickens these sterile-free diets and trying to understand the importance of cholesterol to chickens. And he identified in these chickens a defect in blood coagulation. And this couldn't be rescued by giving fat back to the chickens. It couldn't be rescued by giving cholesterol back to the chickens, but it was rescued by some diets, some seeds, and some leafy vegetables. And so through this line of investigation, he identified a new fat-soluble vitamin called vitamin K, which is named after the Scandinavian or German spelling of coagulation. And so this is where the name comes from, vitamin K. It comes from coagulation, which is spelled with a K in Scandinavian and German languages. It's one good way to remember that vitamin K is necessary for blood coagulation. And I think that this early study really led to an enduring mystery that took many decades to solve, but now we have a pretty good handle of why vitamin K is important for coagulation. But for many decades, it wasn't clear exactly what function vitamin K was playing at a biochemical level, at a mechanistic level, that would be important for vitamin K coagulation, vitamin K and blood coagulation. But we'll talk about why that is today. So vitamin K can come in a variety of different flavors. Shown here is phyloquinone or vitamin K1. These vitamin K share this double ring, this naphthoquinone ring that's methylated, and then each of them have this R group, which will tend to be a branched hydrocarbon chain off of this carbon here. So shown here is vitamin K1. This is going to be the predominant vitamin K in the diets that we get from plants. It's also the form that's found in supplements. Another prominent form is vitamin K2. And similar, again, they share this methylated naphthoquinone ring, that methyl group, and then these benzenes is the quinone rings. And then this is named, this can come in with a variety of lengths in this R group, where here is just shown N equals 4 is menaquinone 4, sometimes abbreviated MK4, and this can come in a variety of lengths. So you might see another one that's common is MK7, but bacteria can make this anywhere from like 1 to like 12 repeats of this length. And so it's easy to see why this would be a fat-soluble vitamin, given that these very long, in some cases it's extremely long, hydrocarbon branch chains, right? They appear a lot like fats, really long fats. So this form is present and synthesized by our gut microbes, and it's also a form that's present in our tissues. So our tissues can convert phyloquinone, this vitamin K1, into vitamin K2. Vitamin K3, menadione, it's just the naphthoquinone ring, the methylated ring. This was used for a long time as a supplement. It was easy to synthesize and purify this, and was used as a vitamin K supplement. And our bodies can take this precursor and convert it into MK4 and use it in our bodies. But it's no longer used as a supplement, at least in humans, because it was observed that high doses of menadione can have liver toxicity. I think it's still present in some animal feeds, but we no longer use this as a supplement. We now only use vitamin K1 as a supplement form for humans. So vitamin K1, phyloquinone is shown here again. It's our primary dietary source. So this is the form we're going to obtain mostly in the diet from leafy plant species. It's also present in vegetable oils. Early work, so Dam's early work on the chicken coagulation suggested that liver, cheese, and eggs would be good sources of this vitamin, but they're actually really poor sources. There is vitamin K in these in liver, cheese, and eggs, and his original study showed that you can rescue the chickens by feeding their coagulation defects by feeding them liver, but these are actually pretty poor sources of vitamin K. So our biggest sources are going to be these leafy plant species and vegetable oils. In this form, phytoquinone and vitamin K1 is what we're going to find in all the nutrient supplements. So in your multivitamin, you'd be getting this form of vitamin K. This other form, vitamin K2 or menaquinone, as I mentioned, it's produced by our gut microbiota. It will be present in small amounts in foods, so in some animal-based foods and some fermented foods, you'll have some of the vitamin K2 contributed by the bacteria in the species. This form is synthesized by our gut microbiota. It's a little unclear how much that actually contributes to our dietary needs of vitamin K, mainly due to the low absorption in the colon. So there's been some research suggesting that it does contribute, that patients that have low vitamin K diets and who are on antibiotics have increased susceptibility to developing vitamin K deficiencies. But this is a little bit controversial, this idea. So I'd say that the amount that's contributed by the gut microbiota is still in question. So as I mentioned, vitamin K is a fat-soluble vitamin, so it's going to go through routes of absorption and transit through our body similar to our dietary fats. So it's similar to eating triglycerol. So it's absorbed in a very similar fashion. So we'll have these fat globules that are broken down and emulsified by our bile acids and bile salts into these mixed micelles, which of course will contain a mixture of digested dietary lipids as well as our fat-soluble vitamins. So vitamin K is one, the others D, E, and A. And these are going to be absorbed across the membrane and then packaged into chylomicrons, just like triglycerols. The chylomicrons are then secreted from the basolateral surface of the enterocyte, and where they can enter into the lymphatic system through the central lacteal, right? And eventually those chylomicrons are secreted into the lymph. This will drain through the thoracic duct into the blood, and then those chylomicrons can circulate, and eventually will be taken up in the liver as chylomicron remnant particles, right? Digested. And so that's the route of delivery for vitamin K from our gut to our liver. And we can see vitamin K in HDLs, LDLs, and VLDLs as well. So they'll be taken up as chylomicrons, but eventually they'll make it to the liver and can be repackaged into VLDLs and secreted from the liver and circulated throughout the body. So the daily recommended intake of vitamin K is around 100 micrograms. The actual storage of vitamin K is rather limited, and this is in contrast to some of the other fat-soluble vitamins, which can really be stored for a long time in the body. And so this 2-day storage of vitamin K necessitates that we get our daily intake of vitamin K every day. And this short time is in contrast to other fat-soluble vitamins. Vitamin K can be stored for a longer time in membranes and to some extent in adipose tissue, but its storage time is on the lower side relative to other fat-soluble vitamins. It can be readily excreted from our body, so there's little toxicity of sort of taking in high amounts of vitamin K. As any excess vitamin K, we can pretty readily excrete from our body, so it's not going to become enriched and concentrated in our fat as some molecules will. So we can excrete this through bile, which would then go out in our feces, or we can secrete this in the urine after conjugation with glucuronic acid. And just recall that the other molecule we talked about that's secreted with glucuronic acid conjugation would be heme, right? So that the bilirubin, biliverdin also can be conjugated with glucuronic acid at the end of the secretion. Okay, so what does this vitamin K actually do in our body? So what is its biochemical function? So vitamin K really has one job, at least one job that we know of. And that's to function as a cofactor for this enzyme called gamma-glutamyl carboxylase, which is sometimes just called gamma carboxylase. And so this enzyme is going to modify glutamate residues. So these are amino acids present in proteins. We see here the amino terminus, the carboxyl terminus, and here's the R group of our glutamate, which has this carboxylic acid, right? And this vitamin K dependent enzyme is going to carboxylate the glutamate, forming a carboxyglutamate, which is sometimes abbreviated as GLA. Right? So glue for glutamate, GLA is for gamma carboxyglutamate. So we have this additional carboxylic acid group added to the carbon. So this is going to increase the overall negative charge on this amino acid residue. So the main biochemical importance of vitamin K is to serve as a cofactor in this carboxylation reaction, converting glutamate to gamma carboxyglutamate. And these are going to become important, especially in coagulation, because they're particularly good calcium chelators, which makes sense because they have these two negative charges, right? Calcium is very highly positively charged. Okay, so carboxylation is going to occur in the endoplasmic reticulum, and the proteins that we know that are carboxylated are secreted proteins. Okay, so here we just have a diagram showing a protein being translated at the ER. So that should look familiar. So we have the endoplasmic reticulum. This would be the luminal side of the endoplasmic reticulum, and this would be the cytoplasmic side. Right? We have here a ribosome that's reading through an mRNA, and it's translating a protein through the Sec61 translocon into the lumen of the ER. And we see two features of this translating protein. It has an N-terminal signal peptide, and that's what actually targets it in the first place. It lets the cell know that this is an ER protein that should be secreted. So the signal peptide is targeting this protein to the ER for secretion. And this is removed usually co-translationally, and then this is followed by a propeptide sequence. Okay, and this propeptide sequence is important because this propeptide sequence is what tells the gamma carboxylase that it should carboxylate these glutamate residues in this protein. Okay, so this propeptide sequence is what binds the gamma carboxylase enzyme. So this enzyme recognizes this propeptide sequence, and then it can carboxylate these proteins. Okay. So we have this translating protein, the signal set sequence is removed. Gamma carboxylase recognizes this propeptide sequence, then together with the help of vitamin K, it carboxylates usually several glutamate residues proximal to the propeptide. And this just shown right here is that the vitamin K is converted into epoxide during this reaction. We'll talk about this in a couple slides when we talk about the vitamin K cycle. Okay, so this enzyme using vitamin K to carboxylate these proteins. These are then moved through the secretory pathway, right? They go out through top vesicles, they go into the Golgi. The propeptide is usually removed in the trans Golgi, and then this protein is packaged into vesicles for snare-mediated exocytosis at the cell membrane, secreting this final carboxylated protein finally into the blood, right? So the end take-home message here is that this proteins go through the secretory pathway and are secreted in a carboxylated state into the blood. And this wouldn't happen if we didn't have our vitamin K, which is required for this reaction. So kind of key points to this carboxylation reaction. We have secretory proteins that are being targeted to the ER by the N-terminal secretory signal sequence. This is usually removed co-translationally. And it's followed by a propeptide sequence which mediates the interaction with gamma carboxylase. Okay, and so this is important because it tells the enzyme gamma carboxylase which proteins to modify. So it doesn't modify every protein, just the ones with this propeptide sequence. So carboxylation of glutamate residues only occurs in select proteins that are indicated by this propeptide. And gamma carboxylase, or gamma glutamyl carboxylase, is the enzyme that converts these glutamate residues into these carboxyglutamate residues. And this propeptide is usually removed in the trans Golgi prior to secretion. Any questions on that? Okay, so this is a little bit more of the biochemistry going on here, which is going to be important. So we have this enzyme, gamma carboxylase, which is mediating the modification of glutamates into carboxyglutamates by the addition of its carboxyl group. And it has a requisite cofactor, and this is vitamin K. And specifically, it's vitamin K in the hydroxyquinone form. This is indicated by the KH2. So you see here that we've lost the double bonded oxygens. Instead, we have these two hydroxyl groups, right, present on vitamin K. And this is the hydroxyquinone form, and it's necessary for this enzyme. During the course of this reaction, vitamin K is modified and converted into epoxide, shown here. And this wouldn't be able to be used in subsequent rounds of this reaction, so we have to regenerate the hydroxyquinone form of vitamin K. And this is done through two reductase steps mediated by the same enzyme, vitamin K epoxide reductase. So it mediates one reductase step, yielding vitamin K. And then it mediates a second reductase step, yielding vitamin K hydroxyquinone. So this enzyme, vitamin K epoxide reductase, is really important for recycling vitamin K for subsequent use in this reaction. So this can be used again and again. And importantly, this vitamin K epoxide reductase is the target of warfarin and coumarin. So these are common blood thinners or anticoagulants. Okay? And we'll come back to this idea as we discuss coagulation. I just want to note that this, if you block this vitamin K epoxide reductase with these anticoagulants, you're no longer able to coagulate blood. Okay, so this is an important medically relevant target. And actually, there's a second reaction shown here with this dimmer kind of gray arrow that can be used when vitamin K epoxide reductase is inactivated to reduce vitamin K to vitamin K hydroxyquinone, and it uses NADPH. Okay? But it only is active if you add lots of vitamin K. Okay? And so that's interesting because when you're on these blood thinners like warfarin, which is blocking this reaction, you can overcome those blood thinners by taking a lot of vitamin K. If you eat a lot of vitamin K in your diet, it's going to counteract that blood thinner you're taking. We'll come back to that when we talk about coagulation. Okay, so take-home messages about this vitamin K cycle. So vitamin K hydroxyquinone, the reduced form, is what is actually utilized. It's the active form that's utilized by the gamma carboxylase. During the carboxylation reaction, this form is converted into an epoxide. So this couldn't be used again in these reactions. So it has to be recycled. And this is done through two reductase steps that is normally mediated by vitamin K epoxide reductase, which is also the target for these blood thinners. The second step, though, can be overcome by high intake of vitamin K because there's another pathway that uses NADPH to reduce vitamin K. So you could overcome this, right? Okay, so let's move on to coagulation. Any questions on biochemistry here, the carboxylation or vitamin K cycle? Okay, so why is carboxylation important? Why is it related to coagulation? So our body uses blood as a liquid transport system for gases and nutrients. It's really an excellent system for pumping gases and nutrients through our body. However, this type of system is prone to springing leaks, right? And so if you have an injury, you can lose a lot of blood really quickly because the circulatory system is under high pressure and it's pumping blood very rapidly. So we need to have response systems to dam leaks and prevent loss of blood, right? So we have a variety of systems in place that mediate this process of hemostasis, essentially repairing any leaks that might spring in our circulatory system. And this occurs through two main systems. There's primary hemostasis, which refers to the aggregation of platelets to form an actual physical plug at the site of injury. And there's secondary hemostasis, and this refers to a activation cascade of coagulation factors, which will eventually form fibrin polymers, which will strengthen this plug. Right? And we can see this shown here in the lighter color. We have the trapped red blood cells or erythrocytes that are trapped in this net-like polymer of fibrin. Right? And so this, importantly, this generates a fibrin polymer or net that will trap blood cells as well as platelets to form this clot. And this involves a set of coagulation or clotting factors. And these factors are the factors that both detect the injury, the site of injury, as well as initiate this coagulation response or cascade. Importantly, there are mutations in several of these factors cause very severe hereditary clotting diseases, so forms of hemophilia. And there's a lot of interest from the pharmaceutical side to address these diseases. And mostly the therapies are around producing recombinant purified factors for injection into the blood. But there's some companies like Sangamo, which is here in the Bay Area, which are working on gene therapy approaches to introduce corrected factors back into the body. And that would essentially cure the disease, which would be really exciting. Several of these factors are vitamin K dependent, require vitamin K dependent carboxylation for their activity. So this is, of course, is the connection with vitamin K and why we have deficits in coagulation in the absence of vitamin K. So a lot of these coagulation factors require carboxylation. And specifically, this refers to factors 7, 9, 10, and prothrombin, which is also called factor 2. And so we'll discuss some of the factors involved in these different coagulation pathways, and we'll try to walk through this so it's not too confusing. Kind of, unfortunately, the way they named these factors was in the order of discovery, which is always a terrible way to name a system, because then the factors aren't actually in sequence of events. And so it's a little bit confusing when we go through those pathways, but that's how they name them. So we're stuck with it. So these four factors, factors 7, 9, 10, and prothrombin, are carboxylated and secreted into circulation. Okay, so they're going to undergo that whole pathway, the ER, the gamma carboxylase, and secretion out into our blood. And so these will be circulating within our blood. And what's important are that calcium ions are recruited to the negatively charged carboxyglutamate residues, and this can bridge the activated enzyme or protein with negatively charged lipids that are exposed during a tissue injury. So here is shown kind of just an illustration of one of these factors. We probably have an active domain over here, and then a domain with several glutamate residues that have been converted into these carboxyglutamate residues, right? So they'll have an increased negative charge on them coming from these additional carboxyl groups. These can then bind to calcium, and chelate calcium, and calcium will serve as a bridge with negatively charged lipid bilayer that's exposed following an injury. Okay? And so we'll get these building and recruitment of these factors to build these coagulation complexes. And we get this because of this negatively charged interactions with calcium sitting here in the middle of this sandwich, right? Does that make sense? So here's an actual image of what that looks like. So this is the illustration, right? Negative charge, calcium, negative charge. This is an actual crystal structure of a lysophosphocerium. So this would be a negatively charged lipid, shown here. And then there's a zoom-in version here. This is the aliphatic, the hydrocarbon chain of the lipid, and then the head group. And then these black circles are the calcium atoms that are sitting at the interface between these GLA residues and the lysophosphocerium. And so what you see here is that for GLA 21 and 17 in this protein, these calciums are sitting directly in association and bridging the interaction between these residues and the phosphocerium head group. Okay. So this is just an actual version of what this would be like, but this is an illustration version. Okay, so eventually this cascade activates thrombin, and this is going to be a really important protease in the coagulation cascade. So thrombin, it's a serine protease, and it does two things. One, it cleaves fibrinogen into the active form of fibrin. Fibrin is what actually polymerizes and forms those net-like structures at the blood clot. And so fibrin is going to, together with factor 13, form these cross-linked, net-linked fibrils. Okay, so factor 13 just helps to cross-link the fibrin polymers. Thrombin also activates platelets, so it's going to cleave a protein on the surface of platelets. And platelets are important because they store proteins involved in coagulation, and as they aggregate, they also secrete a procoagulant phospholipid surface upon which these factors can be recruited. Okay? So all of these together act to initiate this coagulation cascade. And so the ability of thrombin to induce coagulation directly correlates with its carboxylation. Okay? And so thrombin carboxylation is actually a very good measure of vitamin K sufficiency or deficiency. And so this is just a table showing the amount of carboxylated glutamate residues in thrombin. Usually there are 10, corresponding to 100% activity. If you take away 1, you already see a reduction in activity, which is pretty dramatic. But if you take away 2, it's already down to 20%. Okay, so it's very clear. This is a very clear demonstration of how dependent thrombin is on carboxylation. So sometimes it seems like, oh, you're carboxylating 10 of these residues, and it's such a small modification, but it's very important for thrombin activity. Okay, so eventually this blood coagulation cascade leads to thrombin activation, which converts fibrinogen to fibrin. And shown here is structures of fibrin polymers, just showing how these monomers twist together to form these very elongated polymers. And there are going to be 2 converging pathways that regulate these cascades, the intrinsic pathway and the extrinsic pathway. And we'll go through both of these pathways and talk about the relevant components. And so successful coagulation involves amplification cascade of factors. Particularly important, as I mentioned, is factors 7, 9, 10, and prothrombin all require vitamin K dependent carboxylation for their activity, and that this is important because it allows them to chelate calcium, and this is necessary for recruitment to these sites of injury. Okay, so let's take a short break before we go through the pathways. So there are 2 different pathways for coagulation. Eventually these 2 pathways are going to converge to allow for thrombin activation and fibrinogen conversion into fibrin. But there is a series, cascade of events upstream of thrombin activation. And then anytime you see these types of cascades, they're always involved in amplification of the signal, right? So you get the initial signal and then the amplification to initiate this gross response, right? And so the intrinsic pathway responds to exposed negatively charged lipids. Okay, this can also respond to, as I mentioned, platelets will secrete things as well that are negatively charged, which will recruit these factors in addition. It will respond to exposed negatively charged lipids with the initial activation of factor 12 to activated factor 12. So this is usually just indicated by the addition of an A after the factor name. Okay, so factor 12. And most of these factors are proteases, okay, so serine proteases. So this activation usually involves proteolytic cleavage. So factor 12 is going to go on to cleave and activate factor 11. Factor 11 will go on to cleave and activate factor 9. And the factors that I've indicated with circles are the factors that are carboxylated. Okay, so these are going to require vitamin K, right, for activity. So factor 9, this carboxylated protein, is converted to 9A, which can then go on to convert factor 10 to factor 10A, the active form of factor 10, and this is also a carboxylated protein. And this is a particularly important protein because this is the site of convergence of the extrinsic and intrinsic pathways. So factor 10 can finally mediate the activation of prothrombin into thrombin. And then thrombin can do 2 things. One, it converts fibrinogen into fibrin, which can go on to form that net-like insoluble polymer. And then it also goes back and it can promotes the activation of factor 12, and it promotes the activation of factor 8, which is a cofactor for factor 9. Okay, and so these 2 arrows here, coming back to these factors, indicate that thrombin is feeding back, right? And this is positively feedback. So sometimes we'll see negative feedback loops, right? Where a product like heme will feedback and inhibit the upstream enzymes. In this case, we actually have a positive feedback loop. So it's feeding back and further amplifying the cascade. Okay? So we have this amplification step. Let's walk through that one more time. I know it's a little bit complicated. So the initial event will have some sort of injury exposing negatively charged lipids. This could also be aberrantly exposed proteins like collagen. These will recruit and activate factor 12, which sits at the top of the intrinsic cascade. There are a series of proteolytic events. So factor 11 gets activated, factor 9 gets activated, and factor 10 gets activated. Again, annoyingly not in order. And then activated factor 10 can activate prothrombin, again, another protease. And we finally have thrombin, which does two important things. Activates fibrin, and then it initiates this positive feedback loop, further amplifying the signal. Okay? So key events in the intrinsic pathway are the initial exposure of negatively charged lipids. This is going to activate factor 12, and this is also important again because it recruits factors that sit with calcium as this sort of negative charged calcium sandwich. Right? So it's carboxyl residues bind calcium, which combine with these negatively charged lipids. This factor 12, activated factor 12 can then initiate a proteolytic cascade, resulting in sequential activation of 11, 9, and 10. Activated factor 10 finally activates thrombin, which does two things. Converts fibrinogen to fibrin, forming that net-like polymer, and it also initiates a positive feedback loop, so further amplifying this cascade, activating factors 12 and a cofactor, factor 8. Okay. So that, yeah?
What do you mean by
Oh, that makes sense. After I get through the extrinsic. So both cascades are going to utilize a series of separate factors, but they'll both converge on factor 10 to activate prothrombin. Okay, so for the extrinsic pathway, following vascular injury, cells secrete a tissue factor, which is aptly named tissue factor. And this factor is going to bind to factor 7 and promote activation. So you'll have factor 7 activated in complex with tissue factor. And this is then going to directly, this is a shorter cascade, directly activate factor 10. So we get factor 10A, and this is the convergent component that's also activated through the intrinsic pathway, right? Factor 10 then activates prothrombin to thrombin, which can activate fibrinogen, fibrin, and formation of that insoluble polymer. Not shown here, it can feed back into the intrinsic pathway. So in this pathway, it's a shorter pathway. It's called the extrinsic pathway because it involves the secretion of a factor. Right? So the intrinsic pathway you can actually get in purified blood. If you just take blood and put it in a test tube, there are anionic surfaces, so negatively charged surfaces in the test tube, and you'll get activation of the intrinsic pathway. It doesn't require an addition of any additional extra factor. But for the extrinsic pathway, you have to add something. You have to add this tissue factor. It's a secreted protein from the injured cells. Okay. Does that make sense? So extrinsic pathway involves the secretion and addition of an additional factor. So the key events in that extrinsic pathway are that you have tissue factor. It's also known as thromboplastin or factor 3. It's a membrane glycoprotein inside the walls of blood vessels, so it's not normally exposed to blood. But during an injury, it becomes aberrantly exposed to the blood. And during aberrant exposure, it binds to factor 7 and enhances its activation, which can then act on and activate factor 10, which is the convergent component between the two pathways. Okay? Factor 10 is going to go on to activate thrombin and fibrinogen. And importantly, this pathway is a very rapid pathway, and it's likely the main initiator of clotting in vivo. So this extrinsic pathway is probably the biggest initiator of clotting in our bodies normally. But if you take blood and you just have blood in a test tube, it's going to be completely intrinsic pathway. So here are the two pathways shown together, and you can see factor 10 being the convergent component, right? This intrinsic pathway responding to expose negatively charged lipids, having a slightly longer cascade, ultimately culminating in factor 10 activation. Same with extrinsic pathway. It requires tissue factor, has a shorter cascade to get to activated factor 10, right? And we can get this from the diagram. So this is the table of the factors shown here. So I know there are a lot of factors, and it also kind of nicely reflects the function of these factors. Most of these are serine proteases, right? You can see these. Most of these are proteases that are going to act in this cascade of proteolytic events. And I boxed the 4 factors for thrombin, 7, 9, and 10, which you should remember are carboxylated in a vitamin K dependent manner. So for most of these, we use the factor nomenclature, but you can see for the first 3, factor 1, 2, and 3 are actually fibrinogen, prothrombin, and tissue factor. So we normally use more of these common descriptive names, right? But they actually do have these factor names.
Christmas.
They're originally discovered as coagulation factors.
Like the only carboxylated one, or
That's correct. So only the carboxylin ones have a direct vitamin K relationship, but if you don't have carboxylation of these, the whole cascade kind of breaks down. You don't have successful coagulation.
Why is it Christmas?
It's Christmas.
The same injury probably would initiate both cascades. So probably with the most vascular injuries, you'll get exposure of negatively charged surfaces, especially from platelets. So phosphatidylserine is commonly exposed, especially on apoptotic cells, which will initiate the intrinsic pathway. The extrinsic is involved in this exposure of that tissue factor, and that's usually the most robust response for in vivo clotting. So usually when I think in vivo clotting, I think extrinsic pathway, but there's also going to be a component of intrinsic as well. So do you guys see anything missing from this table? Does anything jump out at you? Play that game. What's missing? What's missing from this picture?
6.
Yeah, so there's no factor 4 or factor 6. And so actually, factor 4 is actually calcium. So they no longer use factor 4 nomenclature, they just say calcium. But it was noted for a long time that calcium was required and it wasn't understood why. So the identification of carboxylated residues explained the reason calcium was needed. So calcium is factor 4. And calcium, factor 6 was actually a mistake. That was actually activated factor 5, which they inadvertently called factor 6. And so you no longer have factor 6, but you have this stupid nomenclature where you just lack these factor names, and that's because they made a mistake and called factor 5 activated form factor 6 by accident. I always find these things really funny when you go back to them. So maybe this needs to be renamed. We can drive all the physicians crazy and just rename this all in order of action. Okay, so in patients, as I mentioned, patients are given blood thinners or anticoagulants. Right? These are given to patients that might have a risk of stroke or heart attack to reduce the propensity of them to have form blood clots, right? Which would potentially give you a heart attack or a stroke. And 2 very commonly prescribed drugs are warfarin or dicumarol. The structures are shown here. And these, as I mentioned, block vitamin K epoxide reductase. If you recall from the vitamin K cycle, this reductase, epoxide reductase, is required for vitamin K recycling. Right? So if you don't have vitamin K recycling, you're not going to get carboxylation of those 4 coagulation factors. And so this is commonly given to block. And it's slow acting, around 2 to 3 days. And it's slow acting because when you give warfarin or dicumarol, you already have a lot of these factors circulating through the blood. Right? And these factors, like prothrombin, that are in your blood and they're circulating, are all already carboxylated. So giving warfarin doesn't affect any existing circulating factors in your blood, right? It's only going to affect the newly synthesized factors that are being secreted through the endoplasmic reticulum secretory pathway, right? So it takes a little while to act. So if someone comes in with a heart attack, you probably wouldn't give them warfarin immediately because that's going to take a few days to act. Heparin is a much faster acting drug, and this activates antithrombin 3. I didn't talk about this, but this protein directly inactivates thrombin. And so heparin is going to be a very fast acting drug because it's going to immediately activate circulating thrombin. Okay. In a test tube, often just in terms of blood tests, when they take vials of blood, often they'll either add heparin to prevent coagulation, or they'll just add EDTA, which is going to chelate all the calcium and prevent coagulation. So often when they take the blood, those tubes are have EDTA in them, or they're lined with heparin, and they'll just mix them to make sure to inactivate factors in the blood to prevent any coagulation. They'll remain on the hood. Okay, so vitamin K, so the best known role of vitamin K, and probably for, I think in the 1970s, they recognized that vitamin K was important for carboxylation. This was when the connection with the coagulation factors was made. So probably for the next 30 years, it was thought that this was pretty much the only role of vitamin K. But we've started to discover that there are other proteins besides coagulation factors that are carboxylated by this gamma carboxylase enzyme. And one of them is called osteocalcin. And this contains 3 glutamate residues in the N-terminus that are converted to these carboxyglutamate residues. And this is also known as bone gamma carboxyglutamic acid containing protein, but osteocalcin seems easier. And this is a protein that's secreted by osteoblasts, so cells that give rise to bone cells. And they're required for bone mineralization. So vitamin K deficiency in epidemiological studies has been associated with increased fracture and lower bone density. So for a long time, it wasn't really clear the mechanisms that would connect vitamin K with bone, but osteocalcin perhaps provides one potential biochemical connection between vitamin K deficiency and bone health. And so shown here is just a crystal structure of osteocalcin. Similar to what we had seen in that other crystal structure, it has these carboxyglutamate residues which bind calcium, so it's just shown here in more detail. And this helps bridge the association of osteocalcin with hydroxyapatite crystal lattices. This is extremely abundant in bone. So hydroxyapatite is often called a bone mineral. Right? So it's going to make up a ton of our bone. And so these modified glutamate residues are necessary for osteocalcin to bind this bone mineral. Okay. So a very similar idea to what we saw in the blood is that calcium is bridging that interaction between these negatively charged modified residues and another negatively charged lattice, in this case hydroxyapatite, which is going to have a lot of phosphate, negatively charged phosphate. We'll come back to this when we talk about bone and calcium. The other protein that's been discovered to be modified is this matrix GLA protein. And it contains 5 glutamate residues in the N-terminus that are converted to these carboxyglutamate residues. This is secreted by chondrocytes in vascular smooth muscle. And it actually functions as a physiologic inhibitor of ectopic tissue calcification. So one of the universal connections between these carboxylated proteins in all cases is that they have some connection with calcium, right? In coagulation, we require calcium for recruitment to these negatively charged surfaces. In the osteocalcin example, it requires calcium for recruitment to bone mineral. This matrix GLA protein acts as a physiological inhibitor of ectopic tissue calcification, and it was observed that in knockout animals, when you deleted this protein in mice, they exhibited calcified blood vessels and cartilage, which is abnormal, as well as some bone abnormalities, and these animals died within 2 months. There are actually human patients with mutations in this matrix-joid protein that exhibit calcification of blood vessels as well as bone deformities. Okay. So deficiencies in vitamin K are pretty uncommon in adults. It usually only ends up if there are existing malabsorption syndromes or GI disorders. For example, if you have pancreatic insufficiencies and you had a dysfunction in fat absorption, you would also potentially be susceptible to deficiencies in fat-soluble vitamins like vitamin K. Also, if you're getting a liquid diet, you have to be cognizant of proper supplementation with vitamin K. Deficiencies, however, are more common in infants. So breast milk is particularly low in vitamin K. This can be counteracted if the mother takes vitamin K supplements. It can increase the amount of vitamin K in breast milk. Infant milk formulas usually contain vitamin K supplements. And often newborn babies are just directly administered a vitamin K supplement to avoid any issues with vitamin K deficiency. So deficiency is likely more present in infants than adults. So assessment of vitamin K deficiency is usually measured by examining the percentage of carboxylated protein and looking at how much is non-carboxylated. And usually a good measure is looking at prothrombin. So this will reflect vitamin K intake over longer periods of time, which is useful. So prothrombin has a relatively long half-life, around 60 days. And so similar to when we talked about glucose modification of hemoglobin as a metric of glucose homeostasis for diabetics, this would be a similar measure measuring the modification of prothrombin in the blood, since it has a long half-life. So you can directly measure serum levels of phytoquinone or other forms of vitamin K, but this represents very recent, very acute measurements. So again, be similar to measuring like glucose in the blood, right? It's going to have vast fluctuations depending on diet, right? So that won't reflect sort of long-term intake of vitamin K. Another common measurement that's used is prothrombin time, and this is a measurement of coagulation in vitro. So they take your blood, they take the plasma, and then they add tissue factor to the plasma, and they look at the rate of coagulation. And this has been used for a long time, but it's fallen out of favor more recently because it's not a very sensitive measure. So it has to be a pretty gross vitamin K deficiency in order to have an effect on prothrombin time coagulations. Okay. Any questions? Yeah. Yes, exactly. Okay, let's end here, and Clark has your exams, and I'll